In this video, we're gonna learn about a molecular method to solve the problem of identification of Amphipod species, specifically Gammarus species. Before that, here is the properties of Gammarus species. Gammarus species is actually a diverse group amphipoid crustacean that take part in breakdown process in aquatic ecosystem. Among three different amphipods habitat, Gammarus species commonly found in the freshwater and also marine habitats. So, there must be a lot of species. How to differentiate between those Gammarus species or any differential characteristic? There lies a problem. A few studies has conducted and suggest that the species is so diverse that needed an expert for morphological analysis, and the ontogenic and sexual features not really help to differentiate those species. Therefore, a new study has been conducted which use a molecular method known as randomized amplified polymorphic DNA fingerprinting in identification of amphipods, specifically Gammarus species. So, what is RAPD and how it's been utilized? So, RAPD generally defined as a molecular method or amplification method that used to amplify segment of DNA at random. It uses a random and short primer that anneal randomly low analytic temperature. So this is the working step for RAPD that utilize many short primers. So there are a few advantages of RAPD technique that makes RAPD more better than other molecular technique. Thus, the case study was conducted to identify the potential of RAPD in characterization of amphipod species by determining the species-specific markers. Let's see the method that been utilized in this study, which actually involved three different stages. The first stage would be the amphipods collection and processing. Second is morphological analysis, and third is RAPD procedures. And firstly, for collection of those amphipod sample, it involves a few locations, particularly on the coastal system of Portuguese coast. Then all samples are brought alive to the lab for identification and analysis using stereo microscope. Then 8 adult specimens were selected with equal number of males and females except for 2 species. Then the material that has been examined is listed in this table. Next is the second stages of this experiment, which is morphological analysis that involve 15 specimens from selected Gammarus species with a few important traits such as head and antenna, which further photographed using bright microscope. And the last protocol would be RAPD protocol that involve 3 different processes. The first processes would be DNA extraction that were performed using this conventional extraction method. Next is DNA amplification that was conducted in a final volume of 12.5 ml with this respective primer. Then gel electrophoresis were conducted with these respective steps, and the band that we observed was caught twice using this frequency formula. Then to get the robust or high quality band, this step was performed and all results were poor. Let's move on to see the finding from this study.
So now we are going to look into results. Okay, first is morphological analysis. The features in adult male camarus, including presence of calceolae on flagellum of antenna and existence of angular humps on the dorsal margin of urosome. Rows of city berdasarkan artikel 4 and 5. Apart from that, other morphological analysis including camarus insensibilis. In female, they have the marginated distal margin of coxal platform. And then the number of sepals on the distal margin of the epipeural plates can determine the morphological differences. And the male of camarus species have more prominent secondary sexual characteristic compared to female. The length relation between the outer and inner rami of uropod between three species are varies. Next is about random amplification of polymorphic DNA fingerprints. RAPD DNA fingerprints. PCR fingerprint is to characterize each species and primer according to the bending pattern. But the data provided for some primer were not suitable for run RAPD analysis and species identification. But the clear primer will set bands that are unique for each other species. Camarus species Melita palmata, Camarilla fasciola show their own unique bending patterns. The primer's quality and efficiency aid the differentiation of Camarus. Within Camarus species, the primers like 4 times GACA, OPA10 and OPA9 provide a clear band with unique discrimination and efficiency in species differentiation criteria. So here the figure 2 shows the RAPD bending patterns obtained from Camarus locusta, Camarus insensibilis and Camarus chivrichi with primers of OPA2, OPA9 and OPA10.
Let's proceed to the discussion part. According to the study, the electrophoretic position of amplified DNA bands determine the identification of Camarus species. It can be proved which 9 out of 10 primers exhibited difference of species-specific RAPD profile by using RAPD fingerprint, where each primer revealed a unique bending pattern that holds for each amplified species. The result allowed the identification of the unknown amplified species as G. insensibilis from the previous study by just based on the RAPD fingerprint, which one primer was adequate enough to use as for the identification of that species. For example, the existence of Gammarus species had been recorded from Ria de Alvo, where 10 out of 29 randomly samples of Gammarus were identified as G. insensibilis, while the other remaining species known as G. locusta. However, both of these species had different habitat locations, which G. locusta can be found at higher salinities than G. insensibilis. It is possible to have both species in a similar habitat area, but future study needs to be conducted. However, Mediterranean G. locusta complex were completely unchanged since stocks 1967 in the Taxonomic revision. For example, the findings of the previous study of this freshwater amphipod known as Hyla estica showed that a repeated identification of cryptic species complex in the genetic data. Therefore, a taxonomic re-evaluation of aquatic amphipod as well as genetic monitoring should be maintained and become a standard practice in keeping the life of invertebrates updated. Besides, Karaman and Pinster demonstrated that a universal scoring system for RAPD type 2 would be worth developing as taxonomic identification of forensic insect species. Even though it may be impossible to have an absolute insect identification from this procedure, however, it may be possible to identify the Gammarus species that consist with 120 species, including 105 freshwater species as well as 15 brackish or marine species. In order to improve the technologies of the study, a new combination of taxonomic species with RAPD or other molecule groups have been developed. Firstly, standardizing the procedure by developing the quality controls protocol, including positive and negative control, reference samples, as well as standardizing the materials. Besides, the development of a library of RAPD profile as well as species-specific markers that comprises with morphological features of the species, which allow people to easily access the information. Next, the preserved samples from the previous study should be collected and other ways that can be conducted are by archiving the extraction of DNA sample from reference individuals that can be used as a control for future experiments. Last but not least, the creation of databank of Gammarus taxonomy, including the pictures, morphological descriptions, the preserved parts location, may help people to easily access the information via internet.
In short, taxonomic identification of amphipod species can be identified by using RAPD techniques, and it is an alternative method to morphology-based taxonomic identification. There are some limitations to the conventional method, which include it may show intraspecific variation and it is quite difficult to observe, especially in the examination of few characters. Thus, a simple and universal technique such as RAPD are needed to prevent the loss of relevant information such as diversity, ecology, and distribution of Gammarus species. The advantages of RAPD include: it is a rapid procedure, it can screen large number of loci without advanced knowledge, it can simultaneously process large sample, the specimen tissue needed are low, extracted DNA can be archived and reanalyzed, and it provides consistent markers. Thus, the combination of morphological and RAPD techniques might be very helpful in taxonomic identification of Gamarus species.